Okay, well, good morning, everyone. Nice to see everyone. I hope you had a pleasant holiday weekend. So just a couple of housekeeping things before we dive into the lecture this morning. Perhaps the most important housekeeping item is to remind you, and I included this in the announcement that I sent out over the weekend, that our first term quiz is taking place tomorrow and Friday. So the quiz will open up at 12 a.m. Tomorrow morning. It'll close down at 11:59 on Friday night. And so somewhere within that 48-hour period, you'll need to log in and complete the quiz. So the quiz is focused on the stuff we covered before this week. So the first couple of lectures and the lab we did last week on Daphnia. And so the way these quizzes work, and this is outlined in, I think, the syllabus, is, excuse me, that you will log in. You'll see two questions pop up. So these are two questions drawn out of a pool of 5 questions. So that's going to mix up who gets what a bit. And then you're just going to pick one of those questions to answer. Okay, so don't answer them both, just answer one of the two questions. It's a written answer, just like the practice quizzes have all been kind of short answer practice questions. You have 30 minutes from the time you log in to complete it. If you don't submit it within that time, it will auto-submit. So yeah, those are all the things I think I had to say about the quiz. And then I'll do my best to get that quiz back to you, hopefully Saturday, Sunday, something like that. So you get quick feedback on it, can learn from any mistakes that you make, and just work to keep getting better and better over the course of the semester. Don't also forget that Friday by 5 o'clock is the due date for the first lab assignment. So hopefully you're working away with your group on that. So yeah, don't forget about that. And this week in the lab, we're doing a lab on earthworms. You're going to be dissecting out earthworm guts. You're going to be grinding them up and basically seeing if they have any amylase or protease activity. Are they capable of doing the digestion of starches and proteins, and also whether or not various plants contain compounds that can inhibit those digestive enzymes. So that should be an interesting lab this week. The lecture perfectly pairs with the lab because we're going to spend time here today talking about comparative digestive physiology. And so yeah, let's dive in here. So the first thing I want to talk about here in this lecture, and again, you would have already kind of known this if you thought about it, is that when you think about the kinds of food that animals eat, there's quite a bit of variation, right? Animals eat lots of different kinds of foods, and I have this beautiful sort of collage here, if you will, showing the different types of foods. So we have foods like milk, which of course is a very uniquely mammalian food. We have foods like nectar, right? This liquid that is produced by the flowers of many plants. We have foods like vertebrate flesh, right? So here you see some flies or maybe they're insect larvae of some kind, sort of picking away at the flesh off the bones of this animal. Seeds are a very common food. So here's a bird sitting at a little dish of seed, probably a backyard bird feeder. Whole vertebrates, so here's an owl not bothering to just pick the flesh off of this mouse or rat that it's found, but just literally eating the entire thing. And oftentimes, of course, as you might know, owls won't be able to digest the hair and the bones, and so they'll cough those things back up. And you can maybe you've done little experiments in the past where you get owl pellets, and you have to kind of dig through and see if you can identify the bones that are in there and figure out what the owls ate. Obviously, insects can be a good source of food. Here's a praying mantis, an insect itself, eating another insect. You've got fruit down here. So there's a bat snacking away on these fruit that are, for whatever reason, hanging in some location. Vegetation, of course, by which we basically mean the leaves and stems of plants. And then lastly, detritus. This would include basically the decaying bodies of animals or plants, in addition to fecal matter. So there's lots of different kinds of foods that animals can eat. And what I want to emphasize here is that these foods differ in terms of their quality. So what this word quality means when we're talking about food quality in this context is we're talking about how easily can the compounds that are within these foods, how easily can they be digested, right? Animals, of course, have to ingest their food, and in doing so, they're taking in these big, large, chemicals that are present within the foods they're eating, but they then have to digest them down into smaller, simpler compounds that can actually be absorbed into the body by the gut. So when we talk about food quality, we're talking about how easily can the large complex molecules within the foods that we're talking about, how easily can those large complex compounds be digested, broken down into smaller, simpler compounds. And so what I have here is a figure, and you'll notice that on the x-axis here, it shows all the different food types that I had pictured here in my collage. But on the y-axis, it has this measure, it says proportion refractory. And so what the term refractory means, again in this particular context, is resistant to digestion, right? So it's kind of a quantitative measure here, if you will, of the quality of a food. We're assessing what proportion of the compounds that are in this food are resistant to being digested, refractory. And you'll notice that there is quite a bit of variation in the quality of foods, right? You can see that we have some foods here like nectar and milk and vertebrate flesh that are very low in terms of their proportion refractory. They're very easy for animals to digest. You see things here like whole vertebrates, a little bit harder to digest, and insects too kind of span a bit of a range here, but some insects are pretty easy to digest. There are some that are a little bit harder. And then you get into things like seeds and fruits and vegetation that generally are pretty difficult to digest. So they tend to have these sort of higher refractory ranges. And then, of course, you get to the lowest quality of all the foods, which is detritus, right? Detritus, which we said is kind of decaying animal or plant bodies or feces. This is the lowest quality food there is. I mean, if you think about feces as a good example of detritus, I mean, feces by definition have already passed through one animal's digestive tract, right? So if you're eating that as your food, ultimately you know that a lot of the nutritional content was already presumably absorbed by the animal that deposited those feces in the first place. But if we kind of take a bit of a broad view here of this figure, what we can say then is that the first kind of 4 foods on this list here, or I guess 5, the milk, the nectar, the vertebrate flesh, the whole vertebrates, and we'll lump the insects in there for the most part, they represent high quality foods, right? They have generally lower kinds of refractory values here, right? If we even look at the insects, we're at least acknowledging that some of these insects are really easy to digest, even if there are some that are a bit harder. But nectar, milk, vertebrate flesh, whole vertebrates, insects, we generally consider these to be high quality foods. And by contrast, the other 4, seeds, fruit, vegetation, and detritus especially, we consider these to be low quality foods, right? They have higher proportions refractory, they're harder to digest. And what you might notice here, if you're kind of comparing the high versus the low quality foods, is you might notice that most of the high quality foods, with the notable exception of nectar, are animal-based, right? Most of the high quality foods here, milk, vertebrate flesh, whole vertebrates, insects, these are animal-based foods. And by contrast, most of the low quality foods on the list here, seeds, fruits, vegetation, these are plant-based foods. And so one of the things we can recognize right away is that it's way easier for animals to digest animal-based foods. It's way harder for them to digest plant-based foods. And the reason for this really comes down to the kinds of compounds that are abundant within plants. If you think about compounds like cellulose, very abundant in plants. Think about compounds like lignin, very abundant in lots of plants, especially woody plants. These are compounds that are very difficult to digest. They have chemical structures that make it very difficult for digestive enzymes to access their chemical bonds and break them. And so that's what accounts for the low quality, high refractory nature of plant foods. It's just the kinds of compounds that are ubiquitous and abundant within plants are often of a chemical structured nature that makes them very difficult for enzymatic digestion. And so with this idea in mind that animal-based foods are easier to digest and plant-based foods are harder to digest, you might sit here and think, okay, well, I would imagine then that the vast majority of animals on Earth are probably carnivores, right? That they would choose to eat these high-quality animal-based foods because that would, of course, allow them to maximize how much energy they can acquire from their foods. Right? If you're eating something that's really easy to digest and absorb, you can maximize the amount of energy that you're able to get from your food, maybe minimize the time you have to spend eating and finding food. This seems like a no-brainer. But here is the other side of the coin. Plant vegetation is probably the most abundant food source on Earth, and cellulose in particular, the most abundant organic compound on Earth. So what we quickly then realize is that animals, when choosing the food they want to eat, they're facing this quality versus quantity conundrum. Do you go after animal-based foods that are high in quality, very easy to digest and get energy from, but they aren't as abundant? Or do you go after high quantity, low quality plant-based foods? Harder to digest, but way more readily available. That's kind of the overall, quandary, if you will, that animals are facing. Do they choose high quality, lower quantity foods or low quality, higher quantity foods? And if we try to answer that question, one way to do it is to just look at, well, what proportion of animals on Earth are carnivores versus herbivores? This is a data set, as you can see, where researchers looked at 139 different species of mammals. And they, for each individual mammal, hopefully you can kind of see here that there are these like very thin individual, almost bar plots there that make up this entire figure. But for every species that they included in the study, they measured what percentage of their diet is made up of plants versus animals. And so if we kind of just overlay some information here, what becomes very clear is that pure herbivores, so the mammals that are eating just plant-based foods, are way more abundant than pure carnivores. We probably have twice as many purely herbivorous mammals on Earth than we do purely carnivorous ones. And so that would tell us that when you think about this quantity versus quality conundrum that I introduced, it seems most animals, at least most mammals, are favoring quantity over quality. They're choosing to eat these readily abundant plant-based foods as opposed to eating the higher quality animal-based foods. Now, having said that, you can also look at this figure and realize that omnivory, which of course means you're eating a mix of both plant and animal foods, that is by far the most common kind of feeding type within the mammals, right? We see that there are probably, almost 10 times more omnivores within the mammals than there are either carnivores or herbivores. And so that leads me to the next thing I want to talk a little bit about here in this lecture, which is why is omnivory so common, right? Why is this the most popular, if you will, feeding style in the animal kingdom? And so I want to talk about this kind of from an evolutionary perspective. So I'm going to talk about two studies here that have kind of tried to answer this question of why is omnivory so common. And what you're looking at here is one study where they were looking specifically at mammals. So that's why you see this collection of mammals listed here, the different mammalian orders, all listed here on the left-hand side. Reminds you that this is a mammal-based study. But what they did here is they actually did what's called a phylogenetic reconstruction analysis. They basically looked at all the diets of these modern day mammals, and then they kind of used phylogenetic trees to work backwards and kind of figure out how did these different kinds of diets evolve from the mammalian ancestor to the present day. And so what they were really interested in trying to see through this analysis are these dietary transition rates, right? How commonly within the evolutionary past of the mammals did we see an animal transitioning from one kind of diet to another? You know, from herbivory to omnivory, or from omnivory to carnivory. How often do we see these transitions occur? And so when you look at this figure that's here, you'll notice that this data set here, by far and away, has the highest transition rate. So the most common dietary transition that they saw over the evolution of mammals was herbivores becoming omnivores. This was the most common transition that they saw here in the mammalian evolution, in the evolution of mammals, was herbivores becoming omnivores. The second highest transition rate, which is the blue data set here, is for carnivores becoming omnivores. So right away we have our answer to our question, why do we have so many omnivores? Well, it seems that over the evolutionary history of the mammals, these kind of specialized feeders, carnivores or herbivores, they have been changing over to become more generalist feeders, right? To becoming they've been switching away from these very specialized diets of carnivory or herbivory towards these more general omnivore diets, right? The data here reflect that we've seen these transitions towards omnivory from these more specialized diets. On the other end of the spectrum, if we look at the least common evolutionary diet transitions, so the least common one here appears to be this black bar, right? Carnivores becoming herbivores. And the second least common is this kind of purple bar here, which is herbivores becoming carnivores. This also, I think, makes a lot of sense because, as we're going to talk about here in a moment, you know, being a specialist like a carnivore or a herbivore, it requires a very specialized digestive system, right? And so making the flip from being a pure carnivore with this very carnivore-specific digestive tract to then flipping over to becoming a herbivore, which requires an entirely different kind of digestive tract, as we'll see, that's going to be very difficult to accomplish. It's obviously happened, but just very rarely. So again, when we look at the data we've got here, we see that going from one specialized diet to another, not very common in the history of mammals, but going from specialized diets to omnivory, very common. And that likely explains why we have so many omnivores within the mammals. Yeah. The y-axis is just the number, so posterior probability density. So yeah, it's a measure of the likelihood that transition took place, I think. Can't remember exactly how they calculated the value, but it's basically, in some ways here, you're looking at basically a histogram, right? So you're looking at, how frequently did we see a particular transition rate? So, I don't remember the details on how they calculated it. I wouldn't worry about it too much. It's really the y-axis that's most important here because it tells you it tells you how often we're seeing this transition rate. I can't remember exactly the details of the y-axis, so don't worry about it. I want to show you a similar data set here. This one is for birds. A little bit different structure to the way the data are presented here. But on the outside of the figure, they're showing you these different specialized diet types, right? So frugivores, eating nothing but fruit. Nectarivores, eating nothing but nectar. Piscivores, eating nothing but fish. Carnivores, eating animal-based diets. Insectivores, of course, eating nothing but insects. Herbivores, eating plants. And granivores, eating seeds, right? So you have these very specialized diets. And then in the middle, you have this omnivorous or generalist diet. Here, what you're looking at is the arrows. Which direction are they pointing? And then they give the transition rates on the numbers that are on the arrows. And really what you can see when you look at this figure is that predominantly the arrows are pointing inwards, right? We mostly have arrows pointing from specialized diets towards omnivory, right? That's what we're pretty much seeing throughout. Yeah, you see a few transitions. Here's omnivores becoming insectivores. Here's herbivores becoming insectivores. We do see a few other changes, but really, when you look at the totality of the data, it's mostly arrows pointing inwards, right? So specialist feeders becoming generalist. Most especially, they show here, herbivores becoming omnivores, granivores becoming omnivores, and piscivores becoming omnivores. Those seem to be the three biggest changes. So we're again, just like we saw in the mammalian data, watching animals that had specific diets transitioning to having more generalist diets. Okay, that's exactly the next thing we're going to talk about here. So what causes the transition? Well, the most accepted hypothesis at this point is that it's competition, right? So if you imagine that you are an insectivore, for example, so you eat nothing but insects. Let's imagine that another insectivore shows up in your area, eats nothing but insects. Suddenly now you guys are in this competition for this very specific food resource. You can imagine how that puts selective pressure on an animal to say, oh, now that there's somebody else here also eating the insects that I want, maybe I would benefit from evolving a more generalist diet, from incorporating some other kinds of foods into my diet and therefore become an omnivore as a way to alleviate that pressure, okay? So competition is a big driving force. It doesn't have to just be the competition I described there, where say another insectivore comes in. It could even be just changes in the abundance of these foods. You know, again, let's imagine I'm an insectivore, I eat nothing but insects, but for some reason the insect population in my area starts to dwindle, right? That obviously is going to mean a lot more competition now. This could be intra-specific competition as opposed to inter-specific competition. But if the insect population is dwindling in my area, now me and my other members of my species are all fighting for this very limited food resource. Again, that puts a selective pressure on to say, maybe I have to start figuring out how I eat other things, broadening my diet, becoming an omnivore. So that's kind of the idea. Competition is probably the biggest driver of that transition from specialized diets to omnivory. That answers your question? Perfect. Now, having said that omnivory is a very it is the major kind of animal diet, really, I'm going to spend, I'm going to spend most of the rest of this lecture talking more about dietary specialization, kind of focusing more on the carnivores versus the herbivores, those animals that do consume specialized diets. And where I want to start this conversation is by talking about foraging costs. We have this figure here, as you can see, that's showing us the home range, right, of carnivores, herbivores, and omnivores. Again, this is an entirely mammalian data set. This is where people seem to do most of the work, is on mammals, so we have the most data available. I've got the home range of these animals plotted on the y-axis against their body mass. Obviously, the very first thing you can see here is there's a positive correlation between body mass and home range, and that makes a lot of sense. Obviously, if you're a bigger mammal, you're probably going to patrol a bigger territory, right? Have a bigger home range, so nothing is shocking there. But what they did find in this study is that even once you account for body mass, you can see that carnivores in red here always have much larger home ranges than, say, herbivores here in yellow. And again, I'll draw your attention to the fact that y-axis is a log scale, right? Because again, maybe the difference doesn't look all that big when you're staring at it here, but again, every tick on that chart is a 100-fold change, right? So even though it doesn't look like an impressively big difference between the carnivores and herbivores, we're talking about probably a 50 to 80-fold difference in their home range. So, you know, carnivores are patrolling much, much larger home range territories than similar-sized herbivores do. Now, we think we have a very simple explanation for this. It's because carnivores are eating animal-based foods, which we've already established aren't as abundant, right? So ultimately, if you're eating an animal-based diet and animals aren't as readily available in the environment as a food source, you're going to have to patrol a larger region to find the energy that you need. And so this leads us to recognize that when you think about search costs, right, the energy that is spent to search and find your foods search costs are much higher in carnivores than they are in herbivores, right? Herbivores don't have a hard time finding food necessarily. Plants are pretty much everywhere, right? You can imagine that if I sent you outside of this building and said, I want you to hunt down a plant to eat versus an animal to eat, you'd find the plant to eat in no time, but you might spend a lot more time trying to find animals. Right? I was trying to look today for deer as I was wandering through the back way into campus. Didn't actually see any deer this morning. Saw lots of plants though. Right? So search costs are really high for carnivores because their food is less abundant. Search costs are very low for herbivores because their food is very abundant. Now the other thing we have to appreciate is handling costs, which is how much energy do you have to spend to kind of get the food into your mouth, after you find it. And of course we can recognize again that handling costs are very high for carnivores as well. Right? I mean, here we have a cheetah that's managed to search for and find a gazelle, but the gazelle didn't just go like, you found me, cheetah, and then lie on the ground and wait to be eaten. It takes off. And now you're switching over into these handling costs. Now I have to chase the stupid gazelle, and then once I even catch it, I have to be able to get it to the ground. I got to be able to rip it apart, kill it, eat it. That's not easy, right? So handling costs are also very high for carnivores. Now you could say to yourself, well, why don't carnivores just go after, easier to handle things? Here's a picture of a cheetah, I'm going to say, trying to go after a squirrel. It actually looks more like it's scared shitless of the squirrel and jumping away, but it's going after a squirrel to eat here, a lot smaller, a lot easier to handle a squirrel, but of course you don't get as much nutrition from it. So even if you're a carnivore thinking, well, let's lower my handling costs by going after something easier to handle, you don't get as much of a nutrition reward, right? So you've lowered your handling costs, but you've also lowered the nutritional benefits you derive. Now on the flip side, you might be saying to yourself, well, the handling costs of plants are probably pretty low, right? If I find a plant out in the environment, it by definition cannot just uproot itself and run off and I have to chase it and subdue it. But notwithstanding the fact that plants can't run away from herbivores, the handling costs of herbivores are probably not as low as you would think. And a lot of this comes down to things like many plants have physical protections. Think about this bull thistle here. One of the things I did over the holiday weekend was I was trying to pull weeds out of one of my little garden spaces around the house. And if you're kind of mindlessly pulling the weeds and you grab onto one of the bull thistles, you quickly realize ow, fuck, that hurt, right? These guys are protected with little prickly spines. And so imagine eating this thing, right? If you're a herbivore and you find the bull thistle and you want to eat it, obviously you can't just, you're probably not just going to go in there and put your whole mouth over it and pull it off. You might have to try and work around the spines, or maybe you have to develop some kind of thickened mouth interior so the spines don't, prick you and hurt you. In any case, there's some kind of handling cost to deal with that physical deterrent. But the other thing we can appreciate about plants is that plants also have a suite of chemical deterrents as well, right? Plants make what we refer to as secondary metabolites, which are basically these compounds that are not, they're not for their body, they're not energy molecules, they're these other chemicals that they make that largely serve as deterrents against herbivory. And they've really had no choice. They've had to, as the picture kind of says here, plants again can't run away, so if they don't want to just be sitting ducks for herbivores to come along and eat them, they need to evolve some kind of herbivory deterrents. So some have evolved these physical deterrents. Most plants have evolved a suite of chemical deterrents using these compounds we call plant secondary metabolites. Okay? And these plant secondary metabolites, they usually interfere with physiological processes within the animal body, right? As a way to deter these herbivores from eating that plant again. Again, this actually ties in beautifully to the lab exercise you're going to do today, because we have you as part of the lab this week, again, measuring digestive enzymes of earthworm guts, but then also taking a bunch of plants, things like lentils and coffee beans and spinach, and grinding them up and mixing them with these digestive enzymes, because many of these plants make digestive enzyme inhibitors as secondary metabolites in the hopes that, hey, you will eat me, but I'm going to inhibit your ability to digest me. So maybe you just end up with this huge, massive amount of undigested, unabsorbed food passing through your gut. You end up basically with diarrhea. And so hopefully you think, oh, I'm going to fucking eat that again. Right? Most of us, when we get diarrhea from eating somewhere, we don't go, oh, can't wait to go back there tomorrow. Right? We avoid eating that thing. So this is what plants are trying to accomplish. And again, beautifully fits in with the lab this week. So I want to spend a little time here now talking about kind of these plant handling costs in terms of thinking about these chemical defenses, these plant secondary metabolites. And so I kind of use this flow chart here, if you will, to kind of guide this conversation. So what they do here in this flow chart is they start here, you're a herbivore, and you've encountered a plant that contains a plant secondary metabolite. Now, I changed the order of the lectures this semester compared to previously, just to kind of align with some changes to the labs. In previous years, we've already talked about taste perception at this point. We're not going to talk about this till the end of the semester this time, but what I'll point out here is most of these plant secondary metabolites, you can taste their presence. They taste bitter, right? Most toxic things that you eat taste bitter. Think about the medicines that you eat, or not eat, but medicines you might take. A swig of Buckley's or some other kind of medicine, how bitter these things taste, because medicines are, for the most part, toxic. They're trying to shut down some physiological process in your body that you don't want working. So when you are a herbivore, you can actually tell that the plant you're consuming has plant secondary metabolites in it, and even gauge their concentration because of this capacity to taste them. And again, we'll talk way more about that later in the course. So at this point, you have found this plant you want to eat, you have consumed part of it, you taste the plant secondary metabolites, you therefore have the choice now to say, do I eat this or do I not? If you decide not to eat it, I mean, that's a cost, right? Because now you have this energy-rich plant that you spent time finding that you've now decided you're not going to eat, right? It'd be as if you were to decide you're hungry right now, right? So you walk out and you go down to the Starbucks and you get there all excited to eat and you're like, oh, fuck, the Starbucks is closed. Well, there's wasted energetic investment to go get this thing, find this food source, only to discover you couldn't actually make use of it. So not eating the plant has a cost. If you then eat the plant, there are a couple of things that could happen to that plant secondary metabolite. If again we keep in mind that plant secondary metabolites are generally toxic, if we eat these things, it means we have to be prepared to try and deal with that toxicity, right? Perhaps try to eliminate that toxicity, metabolize that plant secondary metabolite in some way to minimize its effect. And one of the things they highlight here in this flow chart is that some of this modification happens in the gut, right? And I really want to zero in here on this, right? Metabolized by microbes, because most herbivores, and we're going to talk more about this in a second, most herbivores have guts that are packed full with microbes. And these microbes, one of the things they do for the herbivores is they're able to use the pretty complex chemistry that microbes have to metabolize and thereby detoxify these plant secondary metabolites. So you as a herbivore could eat a plant that you know has these toxins in it and then rely upon the microbes housed within your digestive tract to metabolize those plant secondary metabolites into non-toxic forms that won't actually exert much negative impact on the body. So I just wanted to kind of highlight that because it's a pretty important role for microbes in the herbivore digestive tract. On the other side here it says, well, let's let's imagine that these compounds don't get modified in the gut, say by the microbes. They might get excreted in the feces, so they might just pass through and not necessarily get absorbed into the body. Doesn't mean they can't still have an effect. If you think about inhibiting digestive enzymes, I mean, those digestive enzymes are in your gut cavity, so even if you don't absorb these plant secondary metabolites into the body, they could still exert an effect within the gut itself. But if they do get absorbed, then what has to happen is you have to detoxify them. Once they come into the body, where they have the potential to again, exert negative toxic effects, you're going to have to metabolize them in some way, detoxify them in some way. And again, this represents a cost, right? Because they are showing us here in this list all these different kind of mechanisms by which we could detoxify plant secondary metabolites, oxidation reactions, hydrolysis, methylation, et cetera, But of course, in order to do any of those things, you need biochemical pathways with enzymes to do it. And of course, it takes a lot of energy to build and maintain those enzymes. So the detoxification pathways that we can use post-absorption of these plant secondary metabolites represents another very significant energetic cost that herbivores face to deal with the consumption of plants, and in particular, their secondary metabolites. What I want to highlight next here is that generally speaking, given the cost of each of these individual detoxification pathways, there should be no expectation that every herbivore is just operating every single one of these pathways at maximal levels. That would be such an energetic investment as to make herbivory almost untenable, right? It'd be like you saying, well, I'm going to keep every piece of electronics I own on and operating in my house at every minute of the day. All the lights are on, every TV is on, I've got 10 fridges running. Like, you would just go bankrupt paying on the off chance that, well, maybe I'm going to have a party tonight. I'm going to have all these people in here. I need places to put food and all these things. It would make no sense to spend all that energy, just on that hope that maybe it'll pay off in the long run. So what we find generally in plants here is we find that if you're a generalist herbivore, so you're basically eating lots of different kinds of plants, they generally do operate all these different detoxification pathways, but each one operates at just a low level, giving them some basal capacity to get rid of different kinds of plant secondary metabolites that they might encounter in the different plants that they are eating. By contrast, specialist herbivores usually only operate one of these pathways at very high levels. And they operate whatever particular pathway matches the particular plant secondary metabolite that they're going to find in the very specialized plant that they're eating. This is referred to as the detoxification limitation hypothesis. This idea that really, if you're a herbivore, what kind of plants you can eat are kind of limited by this detoxification capacity that you have, right? So again, you could either be a generalist and run all these detoxification pathways at just low levels, right? And that forces you then to say, okay, I can't eat too much of any one particular plant because I can't consume too much of any one particular plant secondary metabolite, because if I do, it might actually exceed my biochemical capacity to detoxify that particular plant secondary metabolite, and I'm going to die. Or I can be a specialist and say, I'll just zero in on one plant that makes this one kind of plant secondary metabolite. I'll jack up the detoxification pathway for that PSM as high as I can so I can get rid of it, but then I just won't bother eating anything else that might contain other kinds of plant secondary metabolites. Let me just show you a little bit of data here that kind of are consistent with this hypothesis, this detoxification limitation hypothesis. Let me introduce you to something here called the bush-tail possum. It's this cute little guy here, and it says it's a generalist herbivore, right? So we know then if it's a generalist herbivore, it's operating all these different biochemical detoxification pathways, but each one at very low levels. The particular genus of plants that bush-tail possums eat is eucalyptus, which is what's shown here. And in this particular study, they were feeding bush-tail possums two different species of eucalyptus. There's the Meliodora here and the radiata. There are two different species of eucalyptus, and each one makes a different plant secondary metabolite. So what they did here in this study is they measured how much of these eucalyptus leaves did these possums eat per day. And what you'll notice is if they were given only the Meliodora species to eat, you can see that they would eat here about 25 grams of that a day, okay? If they were given only the Eucalyptus radiata to eat, they would also eat about 25 grams of its leaves per day. But when they were given the option to consume both, kind of either or, notice that they would eat a little bit of each, but overall now they got up to nearly 35 grams per day. So why were they eating 10 grams more of eucalyptus per day when they could choose both? Well, it goes back to them being a generalist herbivore, right? Because they're a generalist herbivore, they have all these different detoxification pathways at low levels. They don't have any one particular pathway at a high level. So they couldn't eat too much Melliodora when it was the only one available, because if they ate 35 grams of Melliodora, they would likely exceed how much of its particular PSM they consumed, exceed what they can detoxify in a day, and then they would suffer ill effects from it. Same with if they just ate the radiata only. But if they have the option to mix it up and choose both, they can eat a little of column A and a little of column B. They keep that amount within their capacity to detoxify, and then in totality they're able to eat more because they're not risking exceeding the detoxification pathway capacity to get rid of these very specific PSMs that are in these very specific plants. I hope that made sense. Does that make sense? We're okay with that? Okay. The next thing I want to talk about with respect to dietary specialization is to talk, oh, sorry, yeah. Basically, it's a threshold, yeah, right? If you are a generalist, then there's a threshold for every different detoxification pathway, and you can't eat more PSMs than that pathway can get rid of. If you go above that threshold, now you're going to experience toxic effects because you couldn't detoxify the totality of that PSM that you consumed. Exactly, yeah. Okay, I want to talk next here about gut structure, because this is also kind of interesting. So what we're looking at here is drawings of the gut of 5 different mammalian carnivores. And if you look at the drawings of these guts, what you will notice is carnivores have a very simplified gut structure, right? Literally, carnivores have some kind of sac-like stomach, right, just for storing the food that they've consumed. And then they have a very simple tube-like intestine that leads to their anus, right? Very simple gut structure. And this reflects the fact, as we said earlier, that animal-based foods are high quality. They're really easy to digest, so they don't require any kind of gut specialization. A simple, basic gut is all you need. But if we contrast that with herbivores, I have a couple of herbivores here, the kangaroo and the donkey. Notice that the drawings here reflect that there's more complexity to the structure of the herbivore gut. If we look, for example, at the eastern gray kangaroo, you'll notice this is its simple sac-like stomach down here, but then it has this whole elaborated forestomach here. Right? We didn't see this in the carnivores. This elaborated forestomach of the kangaroo gut, in general, we call this a fermentation chamber. And the reason it exists is that it's going to house within it huge numbers of microbes. Now, we just said a moment ago that one of the things microbes are necessary for if you're a herbivore is to help you detoxify plant secondary metabolites. So one could imagine that this kangaroo could eat whatever plant it's eating, and then that food first enters this elaborate fermentation chamber, this elaborate forestomach, and the microbes there have a wonderful opportunity to try and detoxify whatever PSMs might have been present in the food the kangaroo is eating. But here's the other value of fermentation chambers within these herbivore guts and the microbes they contain. It's that these microbes are also really good at digesting molecules that are difficult to digest. You know, especially if we go back to cellulose. There's an abundance of cellulose in plants. It's very difficult to digest. Microbes are often outsourced, really, to do that digestion. So these fermentation chambers that we find in herbivore guts, they are packed full of microbes, and these microbes are not only detoxifying secondary metabolites that are in the plants, they're also assisting the animal with the digestion of these very low quality, high refractory compounds in the plants like cellulose, like lignin, helping to make some of that energy those molecules contain available to the animal. If we look at the domestic donkey, we notice it's got a very simple sac-like stomach, no elaborate forestomach, but where you do see a digestive tract elaboration is in its hindgut. The large intestine of the donkey is very large and elaborate. It's not this simple tube like we saw in carnivores. Plus they have this large structure right where the small and large intestine meet, this large blind pocketed structure here called the cecum. And so the cecum and the large intestine serve as the fermentation chamber in these donkeys. That's where they have all their microbes to help with detoxification and digestion of refractory compounds. And so the thing to take home here is that herbivores have much more complex gut structures than do carnivores. Herbivores always have some kind of elaborated region in their gut, serving as what we call a fermentation chamber that houses an abundance of microbes to help with PSM detoxification and the digestion of refractory compounds. Kangaroos are specifically referred to as foregut fermenters because their fermentation chamber is in the foregut, so kind of where the stomach is. And then donkeys, for example, are referred to as hindgut fermenters because their fermentation chamber is in the hindgut region, the large intestinal region. There's no midgut fermenters, at least not that I know of. The midgut is your small intestine. To my knowledge, there's no herbivore that uses its small intestine as a fermentation chamber. So you have foregut and hindgut fermenters only. What I want to talk next here about is, well, the cost of maintaining that large, elaborate gut in herbivores. And so I'm going to talk about that, ironically, I think, in the context of a carnivore, which is the Burmese python. Burmese python pictured here, as you probably know if you know anything about the way snakes feed, they, of course, are typically eating whole vertebrates. This snake is opening its mouth wide so it can take in this whole rat. And Burmese pythons in particular are kind of cool because they will eat this, say, one large rat, maybe once a month, and then the whole rest of the month they don't eat anything. They just live off the energy they consumed in that one large meal. And so it turns out that this makes them, this makes them an ideal animal model for studying the cost of gut maintenance. Because what you find when you look at the, in this case, the intestine of the Burmese python here, so it says wet mass of the small intestine, and I want to emphasize here that we're talking about the wet mass of the small intestine just itself, not any food or water that might be contained in it, just the mass of the small intestine. Notice here that when they measured the mass of the small intestine in these Burmese pythons over 30 days post-feeding, initially on the day of feeding, before they gave it the rat to eat, the intestine only weighed 16 grams, but within the next couple of days following the meal, they doubled the size of the intestine. And then once they have finished digesting and absorbing the nutrients from that rat, right, they then break their wet small intestine back down to half its size. So this is kind of cool, right? These pythons are basically saying, hey, I don't really need to have a digestive tract all month long because I'm only eating once a month. So I'm going to maintain this very small, minimal small intestine. As soon as I eat, I'll build up the small intestine to twice its size. So I have this increased capacity to digest and absorb this meal. And then once I finish digesting and absorbing the meal, I'll break that small intestine back down and wait for the next meal to come. That's what they're doing. Now, of course, the only reason to do this is because it must be costly to maintain a gut. No animal is going to break down its small intestine and rebuild it unless there's actually a significant cost to just maintaining that gut during times when you're not eating. This observation seems to suggest that there's a significant cost to maintaining a gut. And why pay that cost during those times of the month when you're not eating? Now, Burmese pythons are actually not alone in this kind of interesting feeding habit of eating infrequent large meals, like I just said, having one rat to eat per month. You can see here that boa constrictors and rosy boas and sidewinders do the same thing. But then there are also other snake species that eat more like you and I do. They eat frequent small meals. If you're eating frequent small meals, it doesn't make sense to go through this gut building and tearing down strategy. You're eating small meals all the time. You basically have to maintain your gut structure all the time. Well, you can see here that bull snakes, king snakes, coach whips, racers, these all eat like we do, small frequent meals, and they presumably maintain their gut structure all the time. Well, researchers got very excited because now they said, look, we have a way now to figure out just how much energy does it take to maintain the structure of a gut in these snakes, because if we measure the standard metabolic rate of these different kinds of snakes. Remember, I don't know if you took B34 with me or you're taking it with me now or whatever, but standard metabolic rate is a measure of metabolic rate when you're not feeding, right? That's one of the requirements to measure an animal's SMR. The animal can't be feeding. Has to be in a post-absorptive state, done digesting and absorbing its meal. So the researchers measured the SMR of these frequently eating snakes, right, that maintain their gut structure all the time, and they compared it to the SMR of these other snakes that just eat these large and frequent meals, and who therefore break down their guts when they're done eating. And by comparing the SMR between these two species, the only difference between them really is, do they have a gut or do they not have a gut when I'm making this SMR measurement? And you can see that the ones that don't have a gut, the ones that eat those infrequent large meals, their SMRs were significantly lower compared to the ones that maintain their gut all the time and eat small frequent meals. And so from that difference, we were able to work out that about 20 to 30% of an animal's standard metabolic rate is devoted to just the maintenance of its gut. Right? Think about that, right? Think about 30% of the energy you're spending as you sit here right now could be energy you're spending just to maintain the integrity of your gut so it's prepared for when you go out and eat later. That's a lot of energy. And so right away you can see, hey, if I'm eating infrequently, like those boa constrictors or Burmese pythons, I could save a lot of energy if I tear down my gut in between meals. Let me end with this last point here, which is why are guts so expensive to maintain? It has everything to do with gut epithelial tissues, gut epithelial cells, the cells that line the gut cavity, and how rapidly they turn over. Right? Epithelial cells in general turn over very rapidly because they're exposed to harsh conditions. Right? By definition, epithelial cells are lining body surfaces, lining body cavities. If you think about the epithelial cells that are lining your gut cavity, they're not bathing in some highly regulated solution like, say, your neurons are. They're being directly exposed to all the things in your stomach lumen. They're being exposed to those low acidity conditions when you chug back a Coca-Cola. They're being exposed to, high concentrations of salt when you, gorge yourself on a bag of salt and vinegar chips. They're being exposed even just to the physical roughness of just food and water passing through. So the gut epithelial cells, because they're exposed to these very harsh conditions, they're dying all the time, and you have to do cell division to replace them. In this figure, what they did is they took these gut epithelial cells and exposed them to radio-labeled thymidine. Thymidine, as you know, is a part of your DNA, but not a part of your RNA. So as it gets incorporated into the tissues, it's an indicator of cell division happening. And all these dark regions that you see here, and kind of the upper region of the gut surface, that's where that thymidine was being incorporated in. And so you can see there's a lot of cell division happening there. And consequently, a lot of energy being spent by the animal just to maintain the cellular integrity of its gut. We will end there. And if you want to talk about the things we didn't get around to talking about, for sure we can do that at some point. But for lack of time, we'll stop right there. Enjoy the rest of your day. I'll see you in the lab if you're there this afternoon. If not, I'll see you tomorrow. And yeah, that's all I got.